it. Recording in progress. So, okay, perfect. So we start with lecture 4 and we start with like from very start, like which is about gene cloning. Clear? So gene cloning, like here you can see, gene cloning is basically the concept behind gene cloning is to produce the large quantity of any gene of interest. Clear? And how you produce that large quantity, that you take a gene of interest, we write it as GOI. Gene of interest can be, let's say it can be for any feature. Clear? Gene of interest can be any feature. Let's say if someone is bald and he needs like to grow hair. So you can insert this gene for hair growth into a recombinant plasmid. Remember, recombinant plasmid is acting like a carrier. Clear? Recombinant plasmid is a carrier which will take this, let me change color, which will take this blue gene of interest and it will transfer into the host body. This is
Okay.
just a very introductory or very brief like introduction about gene cloning. Okay. So when
Okay.
gene of interest and plasmid, I told you it's a carrier, and I will explain you in more detail. When they combine, we call it recombinant DNA. Why we call it recombinant DNA? Because it's a DNA from two different organisms. So two different recombining DNA. So this is why we call it recombinant DNA. Clear? And plasmid, we also will use the term vector for this. Okay, so vector is actually the carrier through which like you will get insert gene of interest and you will transfer it to the like different organisms.
So the vector is the plasmid?
Vector is a plasmid carrier.
Okay.
So normally, like what are the 5 steps? I quickly explain you. 5 step is isolation of donor fragment. Let's say I need your hair gene. I will extract that gene. Like maybe I take your hair and grind them, and through all biological techniques, I get your gene for hair. Okay. And then I will choose a suitable vector. Like vector, it is chosen on how it is chosen. Like we choose this vector on the basis of kind of host you are inserting this into. Okay, so second is you choose vector, suitable vector. Third is this step. Third is this step, incorporation of gene of interest into vector. Clear?
Okay.
Like GOI, gene of interest, is incorporated into vector. This is third step. I'm explaining you this because whole chapter is on gene cloning, so at least we should not you only, we should know what are the basics here. We will revolve around these steps and we will go into further detail. So we get gene of interest into vector. Now fourth thing is transformation of recombinant vector. This I told you, this is recombinant. We call it recombinant. It's a combination of gene of interest and vector. Clear? And vector. So it is transformed into host, means you transfer it into the host. Clear?
Okay.
Transformation is step number step number 4. And fifth one is isolation of recombinant host cells. So inside the host, it will go and it will take hold of the host machinery. Which machinery? DNA or RNA. Using that machinery, it will start dividing multiple times inside the host. Clear? So from one copy, you are getting like millions or hundreds of thousands of millions of copies. Clear? So as a result, you amplified your gene of interest. You are amplifying in large quantity of gene of interest. Why we are producing large quantity? Because we don't need this to use for one time. Clear? Like let's say insulin. Insulin, it's not that you only need one dose of insulin. You need repeated dose of insulin. So this way you will produce insulin and you can collect like that insulin in different vials. So you must be taking experimental biology so you know what vial is, like small Eppendorf. You can store in
Yes.
those vials. Clear?
Yeah.
Any questions so far?
What?
Any questions so far?
No.
Okay.
No,
So
clear.
let's quickly go through your slides. Gene cloning is an indispensable molecular biology technique that allows scientists to produce large quantities of gene of interest. Gene cloning links eukaryotic cells to small bacteria or phage DNAs. So eukaryotic cell is gene of interest, and phage DNA or bacteria, it will be vector. In inserting these, so mostly vectors or plasmids. Plasmids are circular DNAs. You remember bacteria, bacteria is circular DNA, which we call plasmid. So those plasmids, they can be used for transferring gene of interest from eukaryote to other organisms and inserting these recombinant molecules into bacterial host cells. So we are using bacterial host cells. Why we are using this, Galia? To produce larger quantity of our gene of interest. Gene cloning can produce large quantities of these genes in pure form.
Okay.
Clear? So what do you need to clone a gene? I just told you 5 steps. What do you need? So you need vector, you need gene of
The
interest.
gene, the vector, and the bacterial host cell, right?
Yes. Yes, you need restriction vector with promoter. Insertion site with insertion site is this is plasmid. So at some position in plasmid, let's say here and here, gene will be inserted. Gene is not like random, it comes and it attaches anywhere. No, there will be complementary sequences to gene. Let's say if here it is A, G, T, C, there will be complementary regions to that gene. Clear? Here. So it will bind to them, insertion site. Then you need restriction enzymes to break this plasmid. So that when you break this plasmid, it becomes open like this. Clear? It becomes open like this, and now your gene of interest, like it can attach here. So sticky ends, where gene of I will explain you like each step one by one.
Okay.
What do you need to clone the gene? Okay, so restriction endonuclease. So number one thing is you need restriction endonuclease. Nuclease, when anything ends with ase, is it is enzyme. Clear? And enzymes mainly break down. Enzyme will, these endonucleases, they will break down DNA into different fragments. Clear? This is DNA. They will cut DNA into two pieces, piece one and piece two. Clear? This is the function of restriction endonuclease. Clear?
Okay.
And they cut, see, this is whole sequence of DNA. They will not cut by their choice or cut randomly. They will cut at a specific DNA sequence. Clear? Specific. There will be some specific sequences where it will cut. It's not anything random.
Okay.
Like let's say E. coli R1. It has restriction enzymes which cut at the sequence G, A, A, T, T, C. C opposite sequence will be what? CTT A G. So restriction enzyme for E. coli, it will cut at this specific sequence. Clear? See, if you read it opposite, it is GAATTC. GAATTC. Are you getting my point?
Yes.
So it cuts at
You
both
mean
ends.
it's
You go this
you
way.
mean opposite?
Yes, they are opposite. See, they are complementary, but they are also see. One is complementary, which G is opposite to C, A is to P, A is to P, T is to A, T is to A, C is to B. One is if you read from here, G is first A T T C, G A T T C. This is not for whole DNA sequences. This is only for this specific region. Clear?
Yes.
So restriction enzyme will now cut at this point and at this point. Okay? So as a result, you will get a DNA fragment like this. Now you will see two fragments. See, when it cuts, let me just put down when it cuts here and it cuts here, what will happen? These two regions will separate. Clear? This will become
Okay.
one region. And this will become,
Those are sticky ends, right?
yes, this will become another region. Clear? And now we have G, C, T, T, A. And now we have A, B, T, C, T. Clear? This, because one enzyme is
Yes.
cutting here, other is cutting here, so it will break them into two. Now these are sticky ends. Clear? It will expose sticky ends. Clear?
Yes.
So restriction endonuclease first discovered in the late 1960s in E. coli. E. coli is bacteria. Are named for preventing invasion by foreign DNA by cutting into. So restriction endonucleases, they are present inside the bacteria. Clear? Let's say this is enzyme. What is the function of this? Let's say this is a foreign particle virus. It enters the bacteria. Clear? When it enters the bacteria, this enzyme will go and it will cut DNA of virus. And when it cuts the DNA of virus, we call it degradation. And it means virus is dead now, killed. Okay? So foreign particle means anything which is coming from outside. So preventing invasion. So virus is trying to invade this bacteria, but bacteria has restriction endonuclease, an enzyme which will cut the DNA or RNA, DNA of virus, and by cutting or killing the DNA, it will kill the virus. Okay?
Okay.
And these enzymes cut at sites within the foreign DNA instead of chewing from them. So it will cut like this. So dividing it into the pieces. By cutting DNA at specific site, they function as finely honed molecular knives. So this is knife, cutting here and here. Clear? Wherever you will get this sequence GAA, TTC, E. coli will recognize and will cut from. Clear?
Yes.
Now we just study one example, which is ECO R1 endonuclease. Now another is HIND3 endonuclease. Their second one is And like we have very, let me tell you, we have different. So they are like E. coli, like E. coli. So hence 3. And they will mostly cut at the sequence AAGCET. And the cut will be at this position, Valia. This way it will cut.
Do I need
Yeah.
to know the sequence?
Yes, no, for now you don't need to know the sequence. As we will move on, we will see like, but it's a
Okay.
simple, like you need to know like there is a specific sequence on which it will cut. For hind 3, it will always be this sequence. Okay? First letter is from
Okay.
the genus age from Haemophilus. Next 2 letters are first 2 letters of the species name in. Haemophilus influenza. D is the strain. Strains are different categories. Clear? There are different
Okay.
categories of hind, like D will be one of those categories. There's a strain is, and the strains they are normally on the basis of, I am not going into that detail, they are normally on the basis of glycogens or different polysaccharide present on the surface of a on the surface of that organism in the cell wall and just leave that thing. So if a microorganism produces only one restriction enzyme and the name with numerical 1 and 1. If more than one restriction enzyme is produced, the num or sequence. So hind 3 means an organism which is producing 3 restriction enzymes.
Okay.
Clear?
Yes.
This is just how you name it. Oh, here we go. So what I told you about hind 3, I told you about hind 3, it is AAGC. So where is it? Yeah, yes, this is hind 3. AAGCT, it means it will cut here. So opposite will be what? T, T, C, G, A, A. So cut will come from here. There. So it will divide into 2 fragments. Those fragments, one will be like, sorry, my bad. One is this fragment, other is, so including this is this fragment. Clear? So you have now restriction endonuclease recognize a specific DNA cutting only a dead sequence. So different sequences. E. coli is where GAATTC. I told you this one as well. There, so you can get to know where they will cut. In exam, they will give you these sequences that E. coli cut at this, and they will give you whole DNA sequence, and they can ask you how many fragments you will get. Let's say if I have a sequence GAATTC, if I have a sequence GAATTC, it means my enzyme will cut here, my enzyme will cut here, so I will get 1, 2, and 3 fragments. Clear
Yes.
or no?
Can you repeat it?
See, if this is whole DNA, I give you a sequence of DNA and I said I have a restriction enzyme ECOR1 which cuts at GAATTC. They'll cut at this point. I will give you whole DNA sequences, and in the whole DNA sequences, there will be hidden sequences GAATTC, GAATTC. It means I have such 2 sequences. E. coli cut at this sequence. My DNA has 2 sequences such. Clear?
Okay.
In a DNA, this is whole DNA. Let's say it is DNA for, it can be DNA for anything. So it will cut here, it will cut here. So as a result, how many fragments you will get? 3 fragments. So
Okay.
you will get 3 fragments of DNA.
Okay.
Got it?
Yes.
Okay. So they recognize 4 base pairs. 4 into 4, 256 base pairs. 6 base pairs they can recognize. They can recognize 8 base pairs as well. 4,
4 is constant, right?
yes. 6 base pairs. We have 6 here. We have 6 here. We have 6. So different, like this enzyme 4, this enzyme 4, 8. Some can recognize 8. Clear? So these conserved sequences are 4, 6, or 8. Clear? They cut DNA reproducibly in the same place. Sticky ends, blunt ends. Clear? So E. coli when cut, it will produce sticky ends. I told you we just discussed like sticky.
I know sticky hands. Okay. So remember, so what prevents restriction in the nucleus from cutting DNA? Any idea? No.
Okay. See, this is bacteria. Okay? And bacteria has a DNA also. But bacteria, I told you, E. coli, let's say E. coli. This endonuclease we get in bacteria. Normally, all of them are from bacteria. Clear? Are you getting my
Okay.
point? Now the question
Yes.
is why this endonuclease is not cutting its own DNA.
Methylation, right?
Yes, bacteria prevent their own DNA from being degraded in this manner by disguising their recognition sequences.
Okay.
What we mean by disguising? See, there are enzymes called methylase. Clear? And these enzymes add methyl groups to DNA sequences, either to adenine or cytosine. Methyl groups can be added to adenine or cytosine. And now
Okay.
when you have CH3 group added to adenine or thymine, it means DNA is modified. Clear?
Okay.
This DNA is now modified. So when this DNA is modified, it means your bacterial endonuclease cannot recognize this and it cannot cut its own DNA.
Okay.
So they are paired with methylases that recognize and methylate the same site, means A or C. Together they are called a restriction modification system. So restriction modification system is, what is restriction modification system? Restriction endonuclease.
Helps us identify the plasmid with the recombinant DNA, right?
No, restriction modification. What? They are paired with methylases. So there are methylases also in the bacteria. That
This is the
recognize,
restriction modification system.
yes, They are together called a restriction modification system. Modification is caused by methylases
Okay.
and the other thing is the methylases is called methylation and preventing from degradation. Clear?
Yes.
Okay. So methylation, and it is providing defense mechanism, defense system. Methylation protects DNA after replication. So a replication is doubling, clear, doubling of DNA. So methylase. After the replication, the parental strand is already methylated. So see,
What happens when the DNA is being cut?
oh, when DNA is cut, you die. Bacteria will die. DNA has all the information.
That's why it
Remember?
doesn't replicate?
Yes.
Okay.
Yes. DNA has all. No, it will replicate. You said it. What you said?
What?
You said that DNA will not replicate.
If it was cut, right?
If it is cut, then you say DNA is no more functional. And even the cell will be no more functional. Because DNA has all the information.
Okay.
This is why when virus gets in, this endonuclease cuts their DNA. DNA is like information center. They cut that and the virus die. Clear?
Yes.
Okay. An early experiment used E. coli to cut two plasmids, small circular pieces of DNA. E. coli is cutting here, E. coli is cutting here. These are plasmids. Tetracycline and streptomycin. This is antibiotic resistance. Like if you will add tetramycin, these plasmids we are getting from bacteria. Clear? So
Okay.
within the bacteria, with this kind of plasmid, if you add tetracycline, it is not going to kill those bacteria. Okay?
Okay.
Like this is how most of the bacteria they become antibiotic resistant. This is antibiotic. I will explain you this. An early experiment used E. coli to cut two plasmids, small circular DNA. This cutting converted circular plasmids into linear. Circular to linear is a straight line. Okay? With the sticky ends. These are the sticky ends.
How is this linear? It's circular.
So later it will become linear, like they will straighten.
Okay.
Yes, you can straighten it. When you are cutting a part of it, it is no more than circular.
Okay.
With the same sticky ends. The ends base pair, some ends recloses with other join the two pieces. Now, two different plasmids, they are cut by same enzyme. Okay? So
Two
same
different
enzyme,
plasmids are cut by the same enzyme?
yes, they are cut by same enzyme, E. coli. Here see, this is enzyme, E. coli, restriction endonuclease. So it cuts at same point, both of them. Okay?
Okay.
So what will happen? They will have complementary DNA sequences, and if you combine them, they will make a recombinant DNA.
Okay.
Do you know why? You know why?
Why what?
Why they will combine?
Because they have the same sticky end, right? Complementary.
Yes, they will have complementary. Yes. So here they will bind because they have complementary ends. Now the enzyme which will join them is after their binding, they're stick together by
DNA.
bonding, like adenine binds with guanine, cytosine. So this binding is by the enzyme we call DNA ligase. Clear?
Okay.
Now recombinant plasmid we get is tetracycline plus streptomycin. It has now antibiotic resistance to both the medicines. Clear? If you are taking any medicine with tetracycline and streptomycin, it is not going to kill that kind of bacteria because now bacteria has antibiotic resistance of both tetracycline and streptomycin. Layers of our
Yes.
summary. Restriction endonuclease recognize specific DNA sequences in DNA molecules and make cuts in both strands. These allow very specific cutting of DNAs. The cuts in the two strands are frequently staggered, so restriction enzymes can create sticky ends that help to link together two DNAs to form a recombinant DNA in vitro. In vitro is inside the lab, outside the cell line. In vitro is anything we are performing outside cells. Like you can say experiment we are conducting in labs. They are in
Okay.
vitro. Now, next thing is after endonuclease you produce sticky ends, and now next thing is vectors. So what I told you, vectors are carriers. Vectors are
They are
what?
the plasmids, right?
Yes,
Okay.
yes, mostly plasmids are vectors. Vectors function as DNA carriers. So gene of interest in vectors, it can be plasmid as well. Okay? Okay. So vector function as DNA carriers to allow replication of recombinant DNA. So they will allow, let's say this is DNA fragment and this is vector. Let's, now what will happen? This DNA combines here. Okay?
Okay.
And we call this recombinant DNA. Recombinant DNA. And then they are combined by ligase. And now you will insert this into host bacteria. Within host bacteria, they will divide like so. Typical experiment uses one vector plus a piece of foreign DNA. Foreign DNA is gene of interest. Clear? Gene of interest.
Okay.
This gene of interest is some function. Let's say this gene of interest is producing insulin. So you need insulin. So you will take out gene for insulin, normal gene for insulin, and insert that gene into a vector. That vector is now transferred to bacteria. Within bacteria, they will divide many times. And when they divide many times, it means you are getting now large quantity of insulin. Large quantity of insulin. So typical experiment uses one vector plus foreign piece of DNA. The inserted foreign DNA depends on the vector for its replication because this why we are into see why we are not adding this directly to the required person because we are going to use it multiple times and for to produce multiple copies we need to insert that into a vector And through vector, it will produce millions of copies of gene of interest. Millions
Okay.
This is the purpose. So the inserted foreign DNA, this is the foreign DNA, depends on the vector for its replication, as it does not have origin of replication. So this for origin of replication is from where replication starts. There. So when you have gene of interest, you don't normally DNA has origin of replication. ORI, origin of replication. There they are recognized by DNA polymerase, and as a result, DNA is replicated. Clear? But when you have
Yes.
gene of interest, you don't have this region of replication. So that you will get from here vector as ORIC, region of replications. Clear? Origin of replications. The recombinant DNA is then, this is recombinant DNA as a whole. Inserted
What
in
binds to the origin of replication?
DNA polymerase recognize those sequences, and from there your replication will start. Otherwise, see, DNA polymerase, it causes one DNA to two DNA replicating. Clear?
Mm.
And how DNA polymerase binds to origin of replication. Origin
Okay.
means start. If there is no point which this
You have a
polymerase
timer?
binds, what?
You added a timer for the session?
Yes, actually one meeting is 40 minutes. So one meeting is about
Because
20. What?
yeah, I know because there is a timer up 1 minute and 50
Yes,
seconds. Or
yes, yes. And we will rejoin the same link.
okay, but for the recording, it will disappear or it will stay.
No, recording after this session, it will save in my like database.
Okay.
And I will send you through like what Google Drive, I think. So because it will be such a large file, I cannot send through WhatsApp or anything like that.
For today, I will only take one session.
Okay. Okay, no problem.
Okay.
So you will end after 1 minute?
Yes,
Okay,
because I
okay.
have a quiz.
Oh, you have a quiz at this time. So we can stop here
Yeah, okay.
and we will
Better
continue.
because I have a quiz.
Did you find it helpful?
Actually, yes, but I think we need to solve questions
Yes,
because
yes.
like the questions are very complicated, especially the written ones.
Send me questions. Do you have questions with you?
No, for I will text you after for the next session. I will try the other tutor on Wednesday, and I will text you.
Okay, okay. Perfect, perfect. Okay, take care.
Okay, thank you.
No problem.